understanding of the problems.
Yeah.
So our next speaker, so thanks so much, JB. Our next speaker will be Roberto Toro, who will tell us about imaging genetics findings of the past, and if imaging genetics traits are heritable, and how we study them.
Hello. So my name is Roberto Toro, and I work at the Institut Pasteur in Paris, in France, and in a lab where we are mostly interested in the genetics of autism, and I'm doing kind of imaging genetics over there. So my background is mostly from neuroimaging to genetics, and presentation is coming. It's a Macintosh presentation.
No, I think my take on that is that you actually need to do exploratory things, but you need to label those as exploratory. And that's the thing. I mean, people usually do exploratory things and then label those with inference, like a statistical inference tool. And that's the problem that we have at the moment. But exploratory analysis are from time to time, they're actually completely necessary. And if you get some results or things that have been like surprising, you need to go back and just duplicate things and so that's the and exploratory analysis are less easy to publish and they shouldn't. It should be like a entirely.
So in the end, also get it down to the imputation of the results, like how you can label them as.
Sorry, didn't get it.
Sorry, it comes down to the interpretation of the results, but if you interpret it as super important or as something that might be side effects there.
Yeah, exactly. Making sure that people cover reflect the significance of your finding.
So I'd really like to emphasize once again the power issues that you made. I mean, this is extremely important and difficult to estimate the power of your study or to estimate the effect size that you have detected. And you pointed out the Witter's curve is obviously something that affects the whole thing. I mean, there's one example, the dopamine 2 receptor in schizophrenia, for example. It used to be a very good candidate gene 20 years ago, and people had some positive findings there, and that could not be replicated because people assumed the effect size that people assumed was too large, and then it was discarded. And now in these large psychiatric genomics consortium, which is similar to Enigma at the psychiatric level, there they find a dopamine D2 receptor once again with a very small effect size in huge samples. So it's also it's dangerous to discard things too early, right? So it's but it's such a difficult field, and I think one has to really feel responsible for the results and not over-interpret things, but really leave open that we cannot make final conclusions at this point, but that would also has to be supported by the journals also, because they want a definite conclusion and they want to have the definite answer, which is often not possible, and they should accept it.
So this is going to be a very long discussion, I'm sure, but Sarah, I think, has one quick point, and then we have to get to the speaker.
Yeah, just on that point with the DRD2 and also with DRD4.
Yeah, you can move back.
So just on that point with DRD2 and then later on with DRD4, those genes have come up, but they're not the variants that people were looking at when they did those candidate studies. So there are many variants within a gene, and the ones that have been popular in the candidate studies are not always the ones that end up coming up when you do the larger powered studies. Yeah.
Okay, so we'll move on to our next speaker, Roberto, and slides are up. So good.
Hi. So, neuroimaging, phenotypes, and heritability. I will start with some just simple definitions. So what we understand by phenotype and genotype. So phenotype is any observable characteristic of an individual. It could be biochemical, physiological, morphological, also behavior, resulting from the interaction of the environment and its genotype. So the genetic constitution of the individual. And then a special type of phenotype would be what we call endophenotypes. So in psychiatry, an endophenotype is an internal characteristic that can be objectively measured. So ideally in a robust and reliable fashion. An endophenotype should be closer to the causative biological processes than an external phenotype. That's what we would like them to be, and which JB was discussing. No, it's not clear that just by measuring something internal, we will be more close, or we will have less genes involved in the phenotype. But that's what we would like to happen. And so David Glann defined an endophenotype ranking value based on both the heritability of the disorder and the heritability of the phenotype and their genetic correlation, which can be useful to prioritize endophenotypes if you have a few. So if you want to go further, these are some interesting references. So now I will start with the part talking about how these phenotypes could be actually encoded in the genome. Okay, and maybe the extreme that we will discuss are between Mendel and Fischer. You know, so what we call the Mendelian architecture, you would have that one phenotype, so the points, I suppose this is the laser. So let's imagine that those are our phenotypes, and we have that this phenotype here is mostly determined by one very focalized genetic source. The cause of this phenotype would be very tightly linked to one specific part of the complete genome. So assuming that this drawing there is the genome. So for example, in the case of SNPs that we were discussing earlier, this would be for single nucleotide polymorphism. It's just the variations in one single nucleotide that will make people in this room different one from another, and it's at around one letter every 1,000 or 3,000 letters. So here it would be this, you know, if like this. So this is kind of more or less 2,000 letters, and one letter difference is what makes you and I different. So what we would like to find are those letters that make this. You know, you have a measurement of your phenotype, and then depending on the genotype that you have for a specific locus, you can have an increase here, an additive increase, linear increase in your phenotype. So you would test for an association, and if your association is strong enough, given the number of SNPs that you are testing, for example, you could establish that there's statistical evidence for this association. But then you have the other kind of extreme of the picture, and reality may be more likely between both, where one phenotype, here again the same, is not mostly determined by one single part of the genome, but more or less by everything in the genome. Okay, so very distributed effect of probably thousands of different genetic variations throughout the genome, every one of very little effect. Okay. So in this case, it's very difficult to pinpoint them. Already, if you were sequencing, because the effect is so small, it would be difficult to figure out which ones are the interesting ones, because even those which are interesting may be just very, very slightly interesting. So how do you study an infinitesimal genetic architecture? Okay, and the way in which people have been doing this for 100 years is by looking at populations. So instead of looking at the genome of people, you suppose that you don't have access to the genome of people, but you have a population, and then you study the variation within a population, knowing, for example, the relationship between siblings, parents, families, twins, etc. So there are many ways in which this can be studied. So normally what you do is you suppose that you have your phenotype, where you can compute a mean phenotype, and then the effect, so the part of the genetic factors would be everything that's related to the genotype once you have taken the mean out. So the phenotype would be a mix between genetic effects, genetic factors, environmental factors, and then the mean value of your phenotype, which again is just a property of your population. It's not a property of the genome of any single person. Okay, so how do you estimate these values? And so here we're decomposing, for example, the variance of the phenotype in one part that would be inheritable and one part that will not be inheritable. This is something very important to keep in mind. For example, the rare de novo mutations, which are by definition non-inheritable, so would come in this account, would be considered as environmental, even though they are genetic. Okay, so it's just the factors which are not inheritable. And then you have a definition of a value which is the heritability of a phenotype, which is the part of the variance in the phenotype that's accounted by the genetic factors. Okay, so the estimation of heritability allows us to study the role of genetic factors without having direct access to them. And there are many different ways in which this heritability can be estimated. So if you can, for example, do breeding, as people do in crops or cattle breeders, you could have this. So let's say that this is a phenotype. Let's say how fast a horse is able to run. Okay, and this is the distribution of the speed of different horses. And now you select the parents for your next horses, and you take those that run the fastest. Okay, so this is the part where you select a certain part of your population. And then you look at what's the speed of the horses that were the kids of these selected parents, and you can eventually see that in average their speed is higher than the speed of the mean original population. So you can estimate here the heritability as the ratio between the response to selection and the selection differential. Okay, this is when you can do breeding experiments, which is not the case for humans. So, but what you can do in humans is you can study the twin cohorts, and this has been very important in imaging genetics practically, and it's probably what most imaging genetic articles are about. So what you have here is the two different types of people. So the monozygotic twins, which share 100% of their genome, and dizygotic twins, which share only 50% of their genome, and we make the rather strong hypothesis that they share a common environment to the same extent. Okay, and what you can do is to decompose this correlation between one part that would be due to inheritable factors, so the genetic factors, and a shared environment. And then you can suppose that the correlation between monozygotic twins will be the addition of those two factors, but then that for the dizygotic twins, they will only share half of those genetic part because they only share 50% of their genome. And so, like this, if your phenotype is strongly inheritable, that would mean that sharing more genome will make your phenotype even more similar. So you can compute the heritability like this. Twice the difference between the correlation between monozygotic and dizygotic twins. And you can also very simply estimate what would be the weight of the common environment, and then what would be due to just a unique environment. And so this is a very simple way of getting very quickly some estimates of heritability, and there are some more accurate ways like the restricted maximum likelihood estimation. Okay, so in the case of height, for example, a classical quantitative trait that we will look more in detail later, this would be like a typical correlation between the height of two monozygotic twins, and this would be the typical correlation of the height between two dizygotic twins. And this, so for height, what you observe is that it's more or less 80% heritability, so 80% of the variation in height is captured by the genome. Now what's very interesting, because today we do have access to the genome of the people, is that this value that I was supposing just because I know how monozygotic and dizygotic twins are, now I can compute it. So based on their genome, I can compute how similar the genome of two people are. And so you can change this value by an alpha that you compute based on the genome. And this is the main idea behind a tool that has been quite producing interesting results in the last years, which is genomic complex trait analysis, or GCTA, by the people at the team of Peter Vischer in Australia. So this was the first paper where the method was introduced. And since then, many people have been able to show that, in fact, there's a large part of the variance in different phenotypes that is actually captured by SNPs when you use them all to compute this genetic distance between people. So here I give just a few examples, but there are many, and the list is growing every week. So, for example, in this first paper, they were able to show that 45% of high diversity was captured by genome-wide SNPs that was using a population of kind of 4,000 people. Now, in the latest paper with a population of 250,000 people, they observed that they can capture 60% of the heritability of height using genome-wide SNPs. For IQ diversity, 51%. Neuroanatomical diversity, our paper recently in molecular psychiatry. But then this is a more like extensive test of different disease-relevant traits by the Yin Yang and the team of Peter Vischer. So from 8 to 77% for 49 different traits, but also so for not only for normal phenotypes, but also for psychiatric relevant phenotypes. So the risk to schizophrenia or Alzheimer's disease or autism is also captured by these genome-wide SNPs. Okay, so one first conclusion would be that human brain diversity and pathology may be encoded by tens of thousands of genomic variants. So there may be a relevance in trying to address this infinitesimal genetic architecture. Okay, so given this fact that it may be important to study a lot of variants which each have a very small effect size, how do we manage to get enough statistical power? And that would depend on your hypothesis. Okay, so I will just briefly consider a few hypotheses. So first of all, if you are thinking about a Mendelian hypothesis, so you're looking for a very concentrated genetic cause, one or a few genes, and then you are looking just at one gene. So you can, in this case, just use a normal analysis of statistical power, and depending on your effect size, and this would be like relatively reasonable effect sizes to test, even though we have the impression today that they may be large. So, but you see that already, even if the effect size you are looking for, it's on the order of 0.2, you will require 800 people, and if it's just 0.1, it would very quickly go to up to 2,000 people. Now, if you are thinking GWAS, not one single gene candidate, but you are looking to all different. So now you can use this type of tool that would allow you, depending on the genetic model that you are using, to compute the statistical power. And these are the values that were just provided as an example by the authors of the tool. And for this, which is actually a fairly large odds ratio, you will require a population of more than 2,000 people. Okay. Now the infinitesimal hypothesis. So in this case, we are not necessarily interested in trying to detect specific genes, but if we just want to know how much of the variance of a phenotype is captured by genome-wide SNPs, we can use this GCTA power calculator. And you can see that if your phenotype is 0.5, which seems to be the case of a lot of quantitative traits in humans, so a heritability of 50%, you will need at least 1,700 people. But if it gets lower, very quickly your population size will have to increase. Now, if you are within the infinitesimal hypothesis, but you do want to find these SNPs, even if their effect is very small, I don't know any tool that would let you do that, but for our paper, we did some simulations. And so running simulations where we suppose that we produce artificial phenotypes where the genes were just one SNP produced all the variants up to 1,000 and then up to 10,000. And so depending on the number of SNPs that were causal, so the ones that you want to find, the different colors show your chances of finding a p-value small enough, for example, to have a genome-wide significant threshold. And if you had even 100 variants in your genome with this population, which was kind of slightly less than 2,000 people, you should be able to find most of the time those variants, which is not the case. So very often we struggle to find one SNP with a population this size, which would mean that in fact the causative SNPs, the causative variants, are much more than 100. We estimated that it's compatible with thousands in our paper. So how to estimate the population size? And maybe the cattle breeders can give us some information. So you have here that the genetic diversity is very small because of the selection. And so cattle breeders have estimated that there's this idea of effective population size. No? So the minimum number of subjects that you need to produce as much genetic diversity as the one you observe. So for cattle, the effective size is about 100 people, 100 animals. And in that case, you would require at least 2,500 animals to predict genetic value accurately. And for humans, the effective population is rather small, but nevertheless way larger than for cattle. So in the order of 10,000 to 15,000 different types of human being. In that case, so to have similar genetic value prediction, we would require populations of the order of 150,000 subjects. Okay, so what we as neuroimagers think is a very large population, maybe well underestimated. Okay, and we can get some information about the size of the population that we need to study if we look at a simpler phenotype. Instead of just starting with neuroimaging, we look at, for example, height. You just need, your giraffe with centimeters or the unit in your country, and you can very accurately measure height in large populations. And so height, as we saw, is inheritable to a large extent, 80%. And here in 2008, studying a combined population of more than 60,000 people, researchers were able to find 54 genome-wide significant SNPs, but they explained less than 5% of the variance. Okay, so which led to the idea of where's this missing heritability? How is that we are not able to find these SNPs, even though we have 60,000 people? So it's interesting to look at the more latest development of the same research program. So now with a population of more than 250,000 people, there appear to be close to 700 SNPs, but they explain 16% of the variance, which is already very, very interesting, and it allowed to do a lot of very interesting analysis of where are those SNPs found, which biological pathways, etc. So it's really something that we would like to do in neuroimaging. But then genome-wide SNPs capture 60% of the variance. So, and the authors estimated that there are probably tens of thousands of causal variants supporting the diversity in height among human populations. Okay, so that gives us an idea, maybe an upper bound for the number of subjects that we will have to study. More references if you want to go further in this subject. There are some very, very interesting, intellectually challenging developments. So for the conclusions up to this point, so human brain diversity may be encoded by tens of thousands of genomic variants, and the detection of these variants may require thousands or even hundreds of thousands of subjects. Okay, and this is the main conclusion from this first part to the study of the neuroimaging phenotypes. Okay, so this fact puts strong limitations on the type of imaging sequencing and the type of analysis we can use. Okay, which have to be accurate, reproducible, and as much as possible automatic. There are many things that we can study in neuroimaging, but they will not scale. To the populations that we require, we have to keep that very simple. So for the moment, that would be structural MRI, so all the different measurements that you can obtain from a T1 weighted image, or from resting state imaging, or anything that you can really make scale. Okay, and I will try to organize the different neuroimaging phenotypes that I will address based on development. So starting from early development, the different parts of the central nervous system are organized in the you can see the organization in the neural tube. So regions which are the most similar among subjects and even among species are those closer to the caudal part of the central nervous system, and then it becomes more and more diverse, more and more different between people and between different species as you move to the more frontal part of the brain. And most of these structures have been shown since very early to be inheritable. This is one of the latest analyses from twin cohorts. So intracranial volume, you see numbers which are very similar to those that we observed for height. So a strong role of genetic factors. So for hippocampus, thalamus, caudate nucleus, etc. This is so. We know now that these phenotypes are strongly inheritable, but are we able to find the genetic causes? This is a first paper from the Enigma consortium by Jason and a lot of collaborators. So they managed to get this very nice population, close to 8,000 people. But then it was, I did a computation at that time. So there were, if you counted all the authors in the paper plus all the people that participated in the consortia, you get like close to 1,200 authors. So it goes back to the normal neuroimaging paper size, no? Kind of 8 people per author. Yeah, and so they were able to find some very interesting information. One SNP associated with hippocampal volume, another one associated with intracranial volume, which points to interesting gene ontology categories that you can see here. But nevertheless, they explain very little variance as JB showed. So this is just the very, very tip of the iceberg. And we will have to go further into increasing the size of our cohorts to get more signal. So this is, again, the latest now paper from the Enigma consortium, as far as I know. So now a population of 30,000 people looking at different subcortical structures, in addition to validating the SNPs that were found previously for hippocampus and intracranial volume. There are new very interesting signals in the putamen and the caudate nucleus. So again, they explain very little of the variance, but they are nevertheless quite strong, so the p-values are very nice, replicated. And from the point of view of the phenotype, so in JSON papers, again, to try to keep the phenotype part scalable, it was just measurements of the volume, which are the most easy to quality control and to get computed in a large consortium. Here, Derek added analysis of the shape of the putamen. So not only the volume, but also the shape. That could be also something interesting that we can just take from the normal T1 weighted imaging. And so they were able to find that the SNP in the putamen was associated with a specific change in the shape of the putamen. So the regions which are more affected by this SNP, so that the width is increasing, are those marked in red. This is so similar to the subject of subcortical structures and brain volume. This is just using the GCTA method, so to try to see the impact of genome-wide SNPs. Our results studying a cohort in the Imagen. So again, because those phenotypes are so strongly inheritable, we are able to do this. No, even if the cohort is not that big, we're able to see that a rather large part is captured by SNPs. So at about 50% of the variance of neuroanatomical variance is captured by the additive effects of hundreds and possibly thousands of SNPs. So most of those SNPs, you are able to, you can also kind of use a subset of the SNPs to compute the genetic distance between people. So you can, for example, take only the SNPs which are within genes and close regulatory regions. And we observed that, in fact, from the variance that was captured from SNPs, 90% of it was captured by SNPs within genes in close regulatory regions. And then that these genetic factors were strongly correlated with those of verbal IQ, but less so for height. Okay, so now going to the cortex. So you know that the cortex is produced by the migration of cells from the ventricular zone to the PL toward the PL surface. So there are two stages. One is the asymmetric, so the symmetric division of the neural progenitors in the ventricular zone that will increase normally the extension of your cortical surface, and then the asymmetric division of those progenitors, so some that will still keep producing more neurons, and those that will become neurons and go to form the cerebral cortex, so increasing the cortical thickness. So probably two different mechanisms to change the extent and the thickness of the cortical surface. And in fact, when Winkler and collaborators looked at the relationship between the heritability of cortical volume, cortical surface, and cortical thickness, they observed that, in fact, even though cortical surface is inheritable in cortical thickness as well, the genetic correlation between both is similar. So they were able to show that their genetic basis for these two phenotypes may be different, orthogonal, which is quite interesting. So, wrong button. This is using a genetic correlation. So again, we are not getting into to the point of specific genes, but we just look if the genes that determine the variability in one point of the cortex are similar to those that determine the variability in another point of the cortex, what's called the genetic correlation. And then they cluster the different regions in the brain based on this genetic correlation. Okay, so here what you are seeing in colors, 1, 2, 3, 4, 5, etc., is the regions with higher genetic correlation, okay, so whose diversity is more likely produced by the same genetic factors. And it produces a very reasonable parcellation of the cerebral cortex. In a later paper, they observed that it's very similar to the fields that you're able to see in mice as well. Now this is like a GWAS approach, but restricted to the genes in the microcephalin family. Okay, so people have known that mutations in these genes produce microcephaly, so a reduction of up to 4 standard deviations in brain volume. And they observed, in fact, that there seems to be normal variation with these same genes seem to be associated with diversity in the extension of the cortical surface in normal populations. Okay, so what's important here is that we were using these cortical surface reconstructions, and it's important to have tools able to produce accurate and automatic reconstruction of the cortical surface. And FreeSurfer is probably the most widely used tool, and you can also obtain several accurate measurements of cortical geometry using MindBoggle, and you will have the URLs in the PDF of the presentation. So now to connectivity, which is more difficult to measure. You would require either functional MRI or DTI. And this is one first example showing the genetics of brain fiber architecture and intellectual performance. So the authors with a very small population, though, for twins, but they were nevertheless showed that the heritability of fractional anisotropy could have values of up to 55%. Okay, and then also you could observe again that there's a genetic correlation between the determinants of fractional anisotropy and those of IQ, which is interesting. So here it would be fractional anisotropy, which is probably the most simple measurement that you can obtain from your DTI, so that you would be able to scale in large populations. So here again, DTI in Enigma is an ongoing project, and so to try to extend these results, but you can follow the work of the Enigma consortium in trying to develop reliable methods for DTI at this URL, if you're interested. And now also, so it's resting state provides an easy way of getting information relatively reproducible from center to center concerning brain function. And this was one of the first papers trying to look at the genetic control of the resting state networks, and with a population of 333, so extended pedigree, a family of cousins, brothers, sisters, etc. And they were able to show that functional connectivity is in fact inheritable. So now we know that most traits show an additive heritability of about 50%. Okay, so probably the first paper that showed that genes involved in functional connectivity are different from those involved in brain anatomy. And this is a more recent result, very interesting again. So here the idea was to look at use information from functional connectivity, but in relation with gene expression data that you can obtain from the Allen Institute. Okay, so first resting state functional functional regions were extracted from a group of 15 subjects. So they extracted the 15 most reproducible networks, and then from those networks they selected those with the least overlap, which are the different networks here. So the different points show the default mode network, the salience network, sensory motor, and visuospatial. And each of the black dots show the points where there's data from the Allen Institute on gene expression. And what they looked was if the genes that are in those regions, their expression profile is more highly correlated. Okay, which appeared to be the case, and it's very interesting. So another way in which neuroimaging phenotype data can be used in combination with data from genetics to obtain information on the relationship between both. So gene expression from the Allen Institute from 6 subjects, not all giving the 2 hemispheres, but and then validation of the results. So the authors were able to check which were the genes that were supporting most of this correlation, and they then looked at Imagen cohort, 259 subjects, and they were able to validate that, in fact, the genes in this top list were those that produce largest difference between subjects in the amount of correlation between these networks. So here you see the group of people in the top and low 20 subjects in relative to the genotype in this list of selected genes, and you see that there's a difference in their functional connectivity. So just for the references, if you want to. So just for the conclusions, and I'm wrapping up with this. So human brain diversity and pathology may be encoded by tens of thousands of genomic variants. And the detection of these variants may require thousands or even hundreds of subjects. And so that really limits the type of imaging sequence and the type of analysis that we can use. Okay, we cannot just do whatever we want. We have to be able to do something very reproducible and accurate and as far as possible automatic. So in these conditions, data sharing, the constitution of large consortia, and the way of mixing this multicentric data becomes essential. We cannot just work on our own. We have to find a way of working all together. And so you can contact me by email, my webpage, and on Twitter. Thank you very much.
If this is a misunderstanding. So you kind of made this distinction between looking at kind of SNPs and looking at this kind of whole genome variants explained. Mm-hmm. So we know that there are differences between the people, and so we would assume there's differences in their genomes. Is there kind of a trade-off between situations in which the phenotypic differences are going to be encoded in kind of infinitesimal differences across a wide number of SNPs, and somewhere you might get actually information about the mechanisms? Or is it actually possible to get kind of information about what SNPs are causing or contributing more in these kind of very broad type of situations?
It's actually very interesting. So most of if you go to this website, there's a summary of all the Mendelian type of mutations. So most of when people are able to find one single gene, it's the disruption of a protein. So really a protein that's not working anymore. But very little of what you find in GWAS would be regulatory. Okay, when you find a large effect. So really I have the impression that the difference that we see here is between the basic makeup of the human physiology or animal physiology, and then what makes us different. So on one side, what makes that we are all the same, that we have 2 arms, that we have normal cognitive functions, and on the other hand, that people are more, Rolling Stones or Britney Spears or whatever. So really what makes us different. And in one case, so if you are able to find, like, for example, it's the case in mental retardation, there are many mutations, strong mutations that are highly penetrant. With just a single one, you have the complete phenotype. And in that case, you can make the animal models, and you can try to do your cell culture and everything to try to get more biological information. When you are in the other case, what it's the complete genome of the person that it's at stake, you will not be able probably to find, and even if you find 15,000 SNPs, what you you are not going to do a mouse model with 15,000 SNPs. So the important information at that point will be the one that we're able to gather. No? It would be, for example, the neuroimaging data that will tell you, look, it's more subcortical, or it's more frontal lobe, or it's more in this network, or this other. So maybe for those cases, the main way of acquiring biological information will be through endophenotypes such as neuroimaging phenotypes, more than through the classical path of making mouse models and cell cultures.
Yeah. I wanted to make actually an encouraging comment. So, because Roberto, you certainly correctly pointed out that there are probably tens of thousands of variants contributing to such phenotypes, and you need hundreds of thousands of individuals. I mean, that is obviously something that is very difficult to practice. And so, an interesting question would be, I mean, what we want is, in the end, to understand the biological basis of these phenotypes that we are studying. How many of the genes or of the genetic variants do we need to know to have a complete picture of the biological processes that are involved? And that may be, so you may need much less than, you don't have to know all the tens of thousands of variants. Maybe, let's say, and that would be the interesting questions, maybe to know 4,000 or 5,000 would be enough to have an impression of the biological pathways that are involved. And all the other genetic variants that are also involved, they fall into the same pathway, the same genes. They are just independent variants. And so I think that will be an interesting question for the future to define and to get experience. How many individuals are really needed? How many genes of the overall genetic variants do we need to know? And this is a little bit encouraging, I think, because maybe we don't need hundreds and thousands of individuals to understand the genetic basis of these phenotypes.
Yep. Yeah. Yep. So probably height is the only example of a quantitative trait where we have a very large population. So with these 250,000 people, they were able to get 700 significant signals, and the amount of data that they were able to get from that was amazing. So you can really see enrichment in different pathways with significant results. You know, for once with small standard error bars, and so you kind of feel confident about So probably if we just have 250,000 people, we will be on business. No, but there's a lot of things that can be said. Even if you are not able to pinpoint which are those SNPs, we can get interesting information. Like I think that the last paper, JB is one of the authors, and what you were able to do with a relatively small population was very interesting. No? So that shows that it's not the only way to go to just increase the number of subjects. Nevertheless, it's extremely important to keep doing it. It's an effort that we have to keep up.
Okay, thanks again.
Thank you.